In 2008, Elaine Mardis' team was the first to sequence the complete tumor and normal genomes of a cancer patient, paving the way for efforts like the Cancer Genome Atlas. GenomeWeb managing editor Kira Curtin spoke with her about what we've learned since, and about the advent of widespread clinical sequencing.
Though genomics is a relatively young field, there have been a lot of advances and changes. What do you think have been the most important events or advances with the biggest impact in the past 25 years?
Well, certainly starting with the biggest one, I would say the Human Genome Project, right? In fact, I think this is the 25th anniversary of the Human Genome Project being sort of completed, and certainly this is a reference genome that's now been improved upon in recent years, but the foundational human genome reference, has been the mainstay of genomic discovery since it was created. So I think, that is a seminal touchstone event. Following on that, there have been a number of other large-scale genomics projects that have really set the foundation for what we understand about, for example, the diversity of human genomes across populations. That's obviously and importantly expanded over time as well, so that we can contextualize findings within different ancestries and within mixed ancestries now as well, leading to more recent notions around polygenic risk scoring and other important aspects of predicting likely common complex diseases as well as rare diseases. And then finally, near and dear to my heart have been large-scale cancer discovery programs such as the Cancer Genome Atlas, the International Cancer Genome Consortium, the Pediatric Cancer Genome Project, and so on. And, again, these have really sort of defined the landscape largely of somatic cancer drivers, dispelled a lot of long-held notions that we had about cancer and how it occurs and how diverse cancers are. And, these are things that we, again, refer to, every day in the work that we do in personalized precision medicine. I think the other thing to say is just that, again, looking backwards, at the time the Human Genome Project was envisioned, at the time large-scale discovery in cancer and other places were, on the run-up, there were, significant detractors who thought that these were going to, take money away from important, small projects, et cetera. And I think in aggregate, what they've done is essentially leapfrogged us forward.
Building on that, I was kind of wondering, when you went into, or when those large programs, the large consortiums were starting, what did you expect to come out of them versus what came out of them? Was there anything surprising that came out that was unexpected?
Yeah, certainly in cancer, where I'm most closely associated over the bulk of my career, have been, numerous surprises, I guess. From a standpoint of historically cancers being described microscopically by pathology, for example, we now understand that the same diagnosis can actually involve a number of different drivers within that diagnosis. We understand that not all cancers are created equal, and unfortunately, cancer finds a variety of ways to, develop and to define itself. I think, one of the surprising things was also that, I think it was thought there were specific genes driving specific cancers, and we now know that's absolutely not the case. In fact, very, very vanishingly few genes would be solely identified with one cancer type. I think that a lot of the different drivers at the time were underappreciated. We've had to revise the hallmarks of cancer because there are now many more. And importantly, I think we also didn't appreciate the importance of the immune system in its interaction with cancers. In the pediatric cancer space, where I do a lot of work now, I think the big learning there was that looking at DNA alone was really not enough. Most pediatric many pediatric cancers, I would say, above and beyond adult cancers, are driven by fusion, oncogenic fusion proteins. And, we can certainly find those in DNA, but it's much more efficient and informative to identify those from RNA, as well as understanding the impacts, because many of these are transcription factor fusions, and so they alter the functional transcriptional programs coming out of those cancers. And then certainly from an epigenetic standpoint, some of the key drivers in the pediatric setting as well as in adults come from a variety of epigenetic regulatory proteins that are altered in ways that, alter the chromatin landscape and determine which genes, that would normally be expressed or not, and vice versa.
And then now, all these things are trying to move into the clinic. So, genetic testing is now fairly common or not uncommon among cancer patients, and there are targeted cancer therapies. What has it been like, both, I guess, doing the work of transferring it into the clinic and also just seeing that it's going that way, that it's that what we've been working on for so long has moved into the clinic?
Yeah, it's pretty gratifying, as you can imagine, right? And that was really the drive to move to Nationwide Children's, because I think you could argue successfully that a lot of the opportunity space is in the pediatric setting. Above and beyond cancer, which is primarily where it's utilized in adults nowadays, we have amazing capabilities to rapidly diagnose patients using whole genome sequencing, advanced analytics, in a 3 to 5 day time period, which is critically important to inform their care, from, a variety of standpoints, whether it's cardiac related problems, general challenges just in terms of aberrant development, et cetera. So I think, that's incredibly powerful on a day-to-day basis to understand the number of lives that we are touching just with our variety of assays that can be performed. And as I mentioned, part of the future forward aspect of this is also just beginning to tally up the variants across the genome that lead to likelihood to develop diseases, not just long-term adult diseases like type 2 diabetes or things like that, which, rely not only on the prediction, but also on modifications to behaviors to be successful in terms of staving off those long-term probabilities.
What are your expectations for the future? Like, how do you think the field might change in the next 25 years? Since we're looking back 25 years, let's also look ahead 25 years.
Yeah, so, in the best case scenario, we'll take some aspects of medicine, as I was mentioning earlier, and turn them from reactive to proactive, right? We'll be able to anticipate and hopefully treat, aspects that can be predicted from the genomic data. The longer term would be that every child will be sequenced at birth, and we can perform a variety of analyses at appropriate times in life and, provide that information future forward. Obviously, one place that's, lacking, but coming up, I think, and there have been some recent nice examples of this, will be a variety of gene therapy approaches for not only addressing from a treatment standpoint, but also for cure. And, we just recently saw the first example of what I would call a bespoke gene therapy that was reported in the New England Journal for a young child with a, single gene defect, highly rare in the population, but definitely repairable using CRISPR gene editing. And that's exactly, what was done. It's not universally applicable yet, but I think there are many, bright, motivated people that are working towards that. So to me, that's one of the longer term, hopefully solutions that we'll have available to us. And then finally, along those lines, also just getting, better medicines in place. Now that we really understand the drivers, we can understand the mechanistic aspects driving some of these diseases. We hopefully can come up with therapies that are suited for those.